So starting off with polysaccharides. Polysaccharides are polymers made by joining many monosaccharide molecules by condensation. Condensation reaction is जब आपके पास loss of water molecule takes place. ठीक है? This is when the loss of water molecule takes place. If we talk about what are the different monosaccharides that we have studied till now, can you tell me?
Um, fructose, no, wait, glucose, fructose, galactose.
Good job. ठीक है. And what is formed when glucose and glucose combine?
Those,
Maltose. Good. ठीक है. So one of the disaccharide we've studied is maltose. What about when fructose and glucose combine?
sucrose,
Good job. And what about galactose and glucose?
lactose.
Wow, you have revised. Nice. ठीक है, good. So these are the three monosaccharides and these are the three disaccharides that we have discussed. Now we are going to have successive monosaccharide joining together by glycosidic bond. And there are many types of glycosidic linkages. For example, अगर आप maltose में देखो, if you have 1,4 glycosidic linkage because the first molecule's carbon 1 and the second molecule's carbon number 4 bind together. That is known as 1,4 glycosidic bond. Got it? This is just known the glycosidic bond, which is the link between the sugar molecules formed by the condensation reaction. Now, the final molecule may be several thousand monosaccharide units long, forming a macromolecule, and the most important polysaccharides are starch, glycogen, and cellulose. These are the three polysaccharides that we are going to study. Can you tell me where have you heard the name of starch before?
Uh, I'm not sure.
Carbohydrates made by the plant are stored in the form of starch. Carbohydrates in humans are stored in the form of glycogen. And cellulose, what about this?
Uh, then plant cell walls.
Good job. They are in plant cell walls. So these three molecules that we are going to start studying, they are the polymers of glucose. Starch and glycogen की बात करते हैं. Since glucose is the main source of energy for cells, it is important for the living organisms to store glucose in an appropriate form. If glucose itself accumulated in cells, it would dissolve and make the contents of the cell too concentrated. This would seriously affect the osmotic properties of the cell and the movement of substances across the membranes as well. Now, glucose is also a very reactive molecule and it would interfere with the normal cell chemistry. So these problems are avoided when glucose is converted by condensation reaction to large polysaccharides. Now, polysaccharides are not exactly sugars because even if you carry out the Benedict test, okay, if you carry out a Benedict test, that is a test for finding sugars, it is only a test, remember, for monosaccharides and for disaccharides. It won't give you a result if you talk about a polysaccharide because polysaccharides are not reactive. Glucose, monosaccharides, small molecules, they were reactive, but when a whole polymer has been made, it's not reactive. It's an unreactive and an insoluble molecule. It is convenient, it is strong, it is compact, it is very strong, but it's unreactive. Why would it be unreactive and strong at the same time? Because it doesn't react easily. It takes so much energy to break all the glycosidic bonds, and no one can, just have that much energy. Now, the storage polysaccharide in plants is starch, and in animals, it is glycogen. So, in those uses, you can already now tell that starch is the storage molecule in plants, glycogen is the storage molecule in humans. And when needed, glucose is quickly made available again by enzyme controlled hydrolysis reactions. Starch is a mixture of two substances, amylose and amylopectin. Now, when we talk about the polysaccharide starch, we have two types in it. One of them is known as amylose, and the other one is known as amylopectin. Now, what's the difference between both of these types of starch mixtures? Amylose is made by condensation between alpha glucose molecules and an unbranching chain of several one-fold linked glucose molecules like this. A long chain would be formed. This is known as amylose. Got it?
Yeah.
Then what is amylopectin? Okay, and they would all be 1,4 linked. They are linked between carbon atoms 1,4 and the chains are curved and coiled up into helical structures like springs as well. But how is amylopectin different? Now this is what? This is amylose, which has 1,4 glycosidic linkages. Amylopectin would also have 1,6 glycogen linkages, which means there's going to be some more glucose molecules over here who will have their carbon number 6 exposed. If you can see that carbon number 6 जो होता है, that is always on the top, right? So if a lot of molecules like this of starch are combining like this and making up a whole chain, amylopectin would have branches as well going from carbon 6. Got it?
Yeah.
If this is an amylose molecule with all 1,4 linkages, carbon number 1,4, 1,4, 1,4, okay, same is the case from carbon number 6, there are going to be more glycosidic linkages as well in amylopectin. They are going to form side branches. So one of the differences, amylose isn't branched. Amylopectin is branched. Amylopectin is also made by 1,4 linked alpha glucose molecules, but the chains are shorter than amylose and they also contain 1,6 linkages. These start branches out to the sides of the chain, and the mixtures of amylose and amylopectin molecules build up into relatively large starch grains. Starch grains are commonly found in chloroplast and in storage organs such as potato tubers, the seeds of cereals, legumes, and they can be easily seen as well. So a polysaccharide is a polymer whose subunits are monosaccharides joined together by glycosidic bond, and glycogen is a polysaccharide made up of many glucose molecules and it is also storage molecule. Here you can see, this is where it was simple amylose. But the moment branches start to be formed like this, okay, this became amylopectin. Got it?
Yeah.
You can see this is the chain of what? Amylose. Okay, if there's only a chain. But the moment this carbon number 6 also forms links with other glucose molecules from here, let's say, okay, they are going to become what? Amylopectin. Amylopectin is a branched structure. Amylopectin has 1,4 linkages and 1,6 linkages, whereas amylose only has 1,4 linkages. Light microscope, especially if it's stained, rubbing a freshly cut potato tuber on a glass slide and straining with an iodine potassium iodide solution is a quick method of preparing a specimen for viewing as well. Starch is never found in animal cell. Glycogen is actually the storage carbohydrate in animals, and starch is the storage carbohydrate in plants. Because starch, when it makes sucrose or glucose in photosynthesis, that glucose or sucrose has to be converted to starch in order to store it. We don't make our own carbohydrates. We have to eat plants. We have to obtain that carbohydrate from somewhere. But the glucose that we obtain has to also be converted into glycogen for storage. It has molecules very like those of amylopectin. Now, glycogen is very much like amylopectin. ठीक है? Amylopectin is there in animals. ठीक है? Amylose is also there in animals. They're both the types of starch. But glycogen is in animals and humans. And it has molecules very like those of amylopectin, which means that glycogen would also have 1,4 glycosidic bonds as well and 1,6 glycosidic bonds as well. And they also clump together to form a huge compact granules which are visible in liver cells and muscle cells as well, where they form an energy reserve. Now, whenever your body is going to face a shortage of glucose, all the glycogen would convert into glucose. But when your body is already high in glucose level, all of the glucose would have to be converted into glycogen. Got it?
Yeah.
Good job. Next polysaccharide and the last one that we are going to study is cellulose. Cellulose is the most abundant organic molecule on the planet, and this is due to its presence in plant cell walls and its slow rate of breakdown in nature. It has a structural role because it is mechanically a strong molecule. Unlike starch and glycogen, the only difference is that cellulose is a polymer of beta glucose. Now, Abhidak, what have we studied till now? Amylose, it is made up of alpha glucose. Then we talked about what? Amylopectin. That is all also made up of alpha glucose, but difference is over here there are only 1,4 linkages. In amylopectin, we had 1,4 and 1,6 linkages. Then we also talked about glycogen, which is also exactly like who? Amylopectin. Now we are going to talk about cellulose. Cellulose is also a structure like amylose. It also has 1,4 carbon linkages, but the only difference is that cellulose monomer isn't alpha glucose. It is beta glucose. Got it?
So amylose's monomer is alpha glucose and amylopectins are made up of alpha glucose, but cellulose is made up of beta glucose.
Good job. Exactly. ठीक है. But the structure is exactly like the amylose. But the only
Okay.
difference is they are all beta glucose. Now, it is a polymer beta glucose, and starch and glycogen are the polymers of alpha glucose. Cellulose is a polysaccharide made from beta glucose subunits used as a strengthening material in plant cell walls. Remember that the OH group on carbon atom 1 projects have the above the ring in beta isomer of glucose. In order to form you remember that what was the difference between alpha and beta? Can you tell me?
The OH,
Yes, now tell me.
like one was above, no, one is below, but I can't remember which one is where.
Okay. Carbon number 1. Look at carbon number 1 of both molecules. ठीक है? Now, alpha is the one where the OH is below the plane in carbon number 1, and beta glucose is the one in which OH is above the plane on carbon number 1. Got it?
Okay.
Now, all of the beta glucose molecules are going to line up together, forming 1,4 glycosidic linkages, and they are going to form the whole cellulose molecules. But the difference is that if this was an alpha glucose molecule, it would link directly with another alpha glucose molecule and then another alpha glucose molecule to make amylose. The problem with beta glucose is that because its carbon number 1 has its OH on the top, right? And the next atoms or the next molecule of beta glucose, these are both beta glucose, right? Now understand this, what I'm trying to explain now. You have seen this is amylose. They are all lined up perfectly, right? They have all formed 1,4 glycosidic linkages. The problem with beta glucose is that this is your molecule A of beta glucose and this is your molecule B of beta glucose. The carbon number 1's OH is above the plane, but the problem is that the next molecule of beta glucose would have its OH below the plane on carbon number 4, right? Because if you look at beta glucose carbon number 4, you can see that it is below, right?
Yeah.
So if a beta glucose has to bind with a beta glucose, how can the OH? Now beta glucose is OH on carbon number 1 is on top, and the next molecule would be touching with the carbon number 4. Its OH is at below the plane. So that is why every alternate molecule of beta glucose is flipped in beta cellulose molecule. Tell me if you've got it. If amylose alpha glucose was forming something like this, ठीक है? They weren't flipped upside down. They were all like in a chain. But beta glucose में क्या हो रहा है? If first is like this, then the next one would be flipped. Is it clear now?
Wait, so why would it be flipped again?
Look, I want carbon number 1 and carbon number 4 OH to be in front of each other. That is why I flipped this beta glucose. Got it?
Oh, okay.
Let me draw it for you. Look. This is a beta glucose molecule. ठीक है? And now I'm going to draw another beta glucose molecule. This is carbon, 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 oxygen, carbon 5. ठीक है? This is, remember, this is your carbon 4, this is your carbon 1, right? So carbon 1's OH is above the plane and carbon 4 OH is below the plane, right? So this is the first molecule of beta glucose and this is the second molecule of beta glucose. They have to link like this. But you can see that in front of OH, we have H. Can the glycosidic bond be made?
Oh, okay.
That is why I am going to flip this molecule so that OH comes over here. ठीक है? This whole molecule is going to get flipped, right? So that OH points towards the OH. So here you can see that every alternate molecule is flipped. Why are we not flipping all of them together? Because that would make the same thing, right?
Then, yeah.
Obviously. So we always have to flip the every alternate molecule. Is it clear?
Wait, if we don't flip it, then there'll be H first.
Then they can't bond. Because in order to remove a water molecule, you need two OH molecules, right? So that water removes and at least one oxygen stays behind. If there's no oxygen behind, you can't form a glycosidic linkages. If you're saying, okay, we leave it like that, we have OH of the first molecule and the second molecule's H is pointing. All right, I can remove the water. But carbon, why would carbon bond with the carbon? We need an oxygen in between to form the glycosidic bond as well, right?
Oh, okay, okay.
Now, a lot of
Wait, so
different
could you
chains. Uh-huh.
wait, so could you go back to the like diagram for a second?
The beta glucose, yeah.
Okay, okay, okay.
Is it clear now? You sure?
Yeah, yeah, yeah.
Okay. So now a lot of cellulose fibrils are going to interact with each other. This is also beta glucose chain, beta glucose chain, beta glucose chain, beta glucose chain. Now a lot of these cellulose chains are going to interact with each other as well. For example, this is one chain. This is going to interact with another chain, then another chain. So it forms a very big crisscross structure that adds to the strength of the cellulose molecule in actual, right? So what happens is now these hydrogen bonds are the ones that actually hold what? The different cellulose microfibrils. This is what we have just talked about. This is one microfibril. A lot of microfibrils are needed, which are going to interact with the help of hydrogen bonds, and between 60 and 70 molecules form tightly crossed links by hydrogen bonding to form these bundles called microfibrils. Microfibrils are then in turn held together by hydrogen bonding by bundles called fibers. Got it?
Yeah.
A cell wall typically has layers of fibers running in different directions to increase the strength. Cellulose makes up the 20 to 40% of the average cell wall. Other molecules help to cross-link the cellulose fibers and some of them form a glue-like matrix around the fibers as well. Cellulose fibers have a very high tensile strength and almost equal to that of a steel because a lot of these beta glucose chains, when they, interact together, they can form a very thick structure. And if pulled at both ends as well, you cannot break or stretch cellulose as well. The high tensile strength of the cellulose fibers makes it possible for a cell to withstand the large pressures that develop within it as a cell result of osmosis. Movement of substances across the membrane that we'll study in chapter number 4. Now, without the wall, what will happen if there is no cell wall? Can you tell me?
Like it will like if water enters, then the cell will burst.
Exactly. Right now, there's a turgidity that can be maintained. The cell becomes turgid right now. It won't when it doesn't. Without the cell wall, the cell will burst when in a dilute solution, and these pressures help provide support for plant by making these tissues rigid as well. They are also responsible for the cell's expansion during growth, and the arrangement of fibers around the cell helps to determine the shape of the cell as it grows as well. Despite their strength, cellulose fibers are freely permeable, allowing water and solutes to reach or leave the cell surface membrane. Got it?
Yeah.
Good. Let me show you one thing. So what do you think is what are the different types of polysaccharides that we have done today?
Starch, glycogen,
Mhm.
amylose.
Good. Amylose is a type of
Um,
this video will The substance that already contains starch, amylose and amylopectin. They are both starches, right?
Yeah.
Good. Was it a good summary? Is it now all clear now regarding just the simple sugars and how many types they are and all?
Yeah.
Good job. Now we are going to talk about dipoles and hydrogen bonds because these are more of a not bonds, you can call these forces. For example, bonds are formed in cellulose, right? These are the bonds which are known as glycoside linkages. Then there's another chain, okay, and then there's another chain. These hydrogen bonds or polar bonds are formed between these chains just to hold them together. We call these dipoles and hydrogen bonds. When atoms and molecules are held together by covalent bonds, they share electrons with each other, and each shared pair of electron forms one covalent bond. For example, if we talk about a water molecule, two hydrogen atoms, each of them would share a pair of electrons with an oxygen atom, forming a molecule with the formula H2O. Now, this is what you can see. Okay? A cell wall is made up of a bunch of microfibrils. And one of the, cellulose fiber contains a bunch of microfibrils. Okay? Cell wall is made from cellulose fiber. This is one fiber, second fiber, third fiber. One cellulose fiber contains microfibrils. And one microfibrils contain what? A lot of beta glucose chains. Got it?
Yeah.
However, the electrons are not shared absolutely equally. Now, what happens is, कि water है, for example, ठीक है? For example, when these cellulose molecules are in water, water structure is polar as well. I think we might have covered this in chemistry or maybe no. Polar means what? When there's a high electronegative element and there are low electronegative elements. As you move towards the right to the periodic table and above the periodic table, the electronegativity increases. So what happens is that the more electronegative element gets a partial negative charge and the rest of the hydrogens both have a partial positive charge. So now when cellulose dissolve in water, water being a polar molecule, what's going to happen? Electrons are not shared absolutely equally because in water, the oxygen atom gets slightly more than its fair share and it has a small negative charge. We know that hydrogen and oxygen have a covalent bond, but the pair of electrons is more closer towards oxygen. Over here, the pair of electrons is also more closer to oxygen. That is why oxygen has a partial negative charge. The hydrogen atom gets a slightly negative fair share. So we call this a dipole. In water, the negatively charged oxygen of one molecule is attracted to the positively charged hydrogen of the another. We call this hydrogen bond. So this is how a hydrogen bond is formed always between O and H. Got it?
Yeah.
So it is all obviously weaker than covalent bond, and this is also something that we will study in your chapter 4 of your AS chemistry. It is much weaker than covalent bond, but still has a very significant effect. You will find out how does it have that much of significant effect. Hydrogen bond is a relatively weak bond formed by the attraction between the group with a small positive charge on the hydrogen atom and another group carrying a small negative charge, for example, between OH groups. Now, hydrogen bond affects many properties of water as well that we're going to cover in the seventh part of your biological molecules. And dipoles can occur in many different molecules, particularly where there's an OH, carbon monoxide, or NH. Hydrogen bond is always formed where? When hydrogen reacts with oxygen, when hydrogen bonds with nitrogen, or when hydrogen bonds with what? Carbon oxygen is also hydrogen bond. ठीक है or fluorine. These are the three four possibilities where you can see hydrogen can react with these as well. Hydrogen bonds can form between these groups because the negatively charged part of one group is attracted to the positively charged part of the another. These bonds are very important in the structure and properties of carbohydrates and proteins. Molecules that have group with dipoles such as sugar. Sugars are all dipoles as well. For example, we discussed water is a dipole. Sugars are also dipoles. And polar molecules are always attracted to water molecules because the water molecules all have dipoles. Sugars, including your monosaccharides and disaccharides, both of them are polar. Polysaccharides are not. Such molecules are hydrophilic. We call these molecules which are polar as hydrophilic because they love water. Because water is also a polar molecule, and those molecules are also polar, so we call them water loving or hydrophilic. And they tend to be soluble in water. Molecules which do not have dipoles are said to be non-polar and they are not attracted to water, so they are also hydrophobic. Now, the ones which are non-polar, they are known as hydrophobic, water hating. So simple sugars, okay, for example, glucose, they are all water loving. We can call them hydrophilic because they do react with water. They do dissolve or mix and touch with water even. But the ones which are non-polar molecules, such as polysaccharides, they are hydrophobic. They hate water. Got it?
Yeah.
And these two terms, hydrophilic and hydrophobic, are actually very important for your जो आपका chapter 4 है, the cell membrane and transport. We are going to study that these two terms again in that. And that is all for the carbohydrates. ठीक है? This was a big topic including monosaccharides as well, disaccharides as well, and polysaccharides as well. ठीक है? Let me show you one more thing. So can you tell me what is the difference between polar and non-polar? What do you think?
Um,
Mhm.
Not so sure.
What do you think polar means? Charged?
Yeah, wait. Uh,
Yeah, you can do it. Yes.
yeah, polar has charges and non-polar
Good. ठीक है.
doesn't have charges.
Simple as that. And how does something become polar then? Anything can become polar the moment it has one of the atoms having a partial negative charge and one of the atoms having a partial positive charge. Got it?
Yeah.
Hi guys, they're long, complex carbohydrates, and we call them polysaccharides because they're made of many saccharides. So some polysaccharides can be found in certain foods, for example bread. We can find them in rice and various types of pasta as well, and they're a very important carbohydrate. So a polysaccharide is basically a polymer of carbohydrates made up of many monosaccharides. So remember, the simplest form of a carbohydrate is a monosaccharide. And when we join lots of these up as monomers, we end up with one long chain, which we describe as a polysaccharide. Poly meaning many. Remember that 2 would be a disaccharide, but more than this is many, and we just name it as a polysaccharide. We tend to say that in order to be a true polysaccharide, they need to have more than 10 monosaccharides joined together into a chain. Any smaller chains than 10 tend to be called oligosaccharides. So these kind of terms can be seen in lots of biological molecules. Mono always means 1, di means 2, oligo tends to mean about 3 to 10, and poly is more than 10. So in this case, we have 8 monosaccharides, so this would be an oligosaccharide. So it does depend on how many there are in terms of what we call it. And saccharide is referring, of course, to carbohydrates. So obviously you can see these prefixes in lots of types of biological molecule as well, like proteins and nucleic acids. But in the case of carbohydrates, we're talking about saccharides. So in order to create these polysaccharides, we form them by joining together monosaccharides in a series of condensation reactions. So remember, condensation reactions are what join monomers together to perform polymers. So each time a monosaccharide is added to another monosaccharide, also a growing chain, there's going to be a condensation reaction. So we might start off first with 2 monosaccharides, and as they come together and are joined by a condensation reaction, we end up with 2 monosaccharides joined together into one unit called a disaccharide. And then to increase the length of this chain, we just need to simply increase it by adding another monosaccharide. And again, a condensation reaction adds this extra monosaccharide to form this now trisaccharide or oligosaccharide. Again, this can then be added with more and more and more until they keep growing in size to form a polysaccharide. But as you can see, each time a monosaccharide is added, it's a condensation reaction adding on a unit every time. In the reverse of this, polysaccharides are broken down in the reverse of condensation, which is hydrolysis. And there's a series of hydrolysis reactions to form individual monosaccharides. But this can happen in a couple of different ways. Let's say we start off with this polysaccharide or oligosaccharide, and we want to break it down into its 4 individual monosaccharides. We can do it either by snipping off one monosaccharide at a time from a chain and going in this direction. And every time this happens, we have a hydrolysis reaction. And then in the end, we would be left with 4 monosaccharides. Alternatively, we can do it a different way, whereby the oligosaccharide is snapped in half to give 2 disaccharides. And then each of these are broken down into their individual monosaccharides, again to give 4 monosaccharides. But the main point is, either way, it's always done by hydrolysis reaction, snipping off monomers at a time. They're different to monosaccharides and disaccharides because polysaccharides are not sweet tasting and they're not very soluble, so they don't dissolve very well in solutions. So they're not what we call sugars, whereas disaccharides and monosaccharides were. But polysaccharides come in much more variety, and the reason is because of the following. We can change the type of monosaccharide that we incorporate into a polymer. So for example, say we chose alpha glucose as the monosaccharide. If we were to add that into a chain into a polymer, we end up with a molecule called amylose. If we incorporate beta glucose, the other isomer of glucose, we end up with a completely different polymer known as cellulose. And just by changing the monosaccharide by a tiny amount, the actual structure of the overall polysaccharide and the properties are very different. We can also change not only the monosaccharide, but how the monosaccharides bonds together. So for example, in both of these cases, we have alpha glucose as the monosaccharide, but in this case we have a particular type of bonding called 1,4 glycosidic bonding, and this gives us amylose. But in this case we have 1,4 and 1,6 glycosidic bonding. Don't worry too much about what 1,4 and 1,6 mean because we'll get onto this. But by having different types of bonding present, we can make different types of molecules. And this one is amylopectin. So by changing different features of polysaccharides, they can get multiple different properties and have very different functions. Polysaccharides are important energy stores for both plants and animals. Plants and animals need to carry out respiration, and the main source of this is alpha glucose. It's the main source of energy for any respiration in any cells. So a molecule of glucose is combined with a molecule of oxygen. In respiration, these produce the waste products CO2.
So this is how you should also know that these monosaccharides are also used in respiration. Got it?